I'm Daniel Levine, and this is the Bio Report. While AI has been seductive in its promise for revolutionizing drug development, one of the constraints remains the quality of the data that's used by any given platform. BPG Bio, an early innovator in the application of AI to drug development as Berg, is taking what it calls a biology-first approach. It capitalizes on its proprietary biobank to conduct multi-omics analysis to understand the biological mechanisms of diseases. We spoke to Niven Narain, CEO of BPG Bio, about how the company's platform technology uncovers novel targets, its evolution from its start as Berg, and how the platform continues to provide insights into experimental therapies after they advance to the clinic. Niven, thanks for joining us.
Well, it's great to be on with you.
We're going to talk about AI and drug discovery, BPG Bio, and its therapeutic pipeline. Let's start with the well-known challenges of drug development, which continue to be expensive and require a long time frame. What specific challenges in discovery and development do you see your AI platform addressing?
Well, first of all, Daniel, I think that as we all look to the future, we're seeing a tremendous rise in brain health diseases, aggressive solid tumors, and of course, metabolic diseases. And at its core, the biggest issue is that we still, as of yet, do not fundamentally understand the real triggers in the biological processes that drive these diseases. Now, we've made a significant headway in some cancers, in diabetes, and cardiovascular disease. But really, what BPG is focused on is taking a step, backwards into the biology. So before we even seek to discover, develop drugs, what we're really doing is starting our process with human tissue samples and annotated biofluids and health records and demographics. So what we're really trying to do is construct a story and a narrative on the individual patient. And as you can imagine, as we then stack this in a manner that allows us to understand a group of patients with a given disease versus a non-disease group, we're able to really use the AI to help us understand areas in the patient's biology that seems to be deviant or different or dysregulated, and those are really where we start to shine this light on the biology that's different in a disease population versus a healthy population, and that's really what separates us and in how we think of drug development, but more importantly, how do we actually perform the drug discovery.
We first spoke to you on the show back in 2014 when BPG Bio was Berg,
Yes.
an early innovator in bringing AI into drug discovery. Can you talk about the transaction that transformed Berg into BPG Bio? And did this just reflect a change in ownership, or was there something more substantial that changed in the company's approach?
It's really a few things. So I'll start up by saying that first of all, the transaction from at BPG that essentially did an all-asset sale of the intellectual property and the technologies and the know-how, along with its talent from Berg. Then essentially, what was the underpinning and the foundation of what is BPG today, and the real impetus and catalyst that drove that transaction was that Berg had pioneered the early discovery processes around AI in drug discovery, right? We were, one of the first groups, if not the first group, to hold, the early patents on the use of Bayesian AI and patient models for drug discovery. And then we really spent a number of years validating that interrogative biology platform, which is today the NI interrogative biology platform. And then from there, we wanted to show that we could be able to develop drugs, or we can be able to uh, identify early disease biomarkers. So in the journey of drug development, we wanted to be able to not only demonstrate that we can develop and advance a drug, we wanted to do it in a way that was patient centric. And really, the how we achieve that is to also look at components in diagnostics and biomarkers that would want to help us understand how patients were different, help us to perform, trials in a more defined and specific way, but also help us to understand, who may be responded to a drug or not. So along the development pathway, we really use the technology to uh, help to unravel these novel insights into biology, so that we can accelerate and define drug development in a more patient centric and efficient manner. And the transaction up to that point, having of course, like, weathered the storms of the pandemic, we were able to develop a pipeline of assets in oncology, and rare inflammatory diseases, like childhood diseases like epidermolysis bullosa, aggressive solid tumors like GBM and pancreatic cancer, and then early assets in neurodegenerative diseases like Parkinson's and Huntington's. And really, what the transaction for BPG, it really helped us cross that threshold from being an early discovery and development company into a pre-commercial company. So since January of 23, the institution is really focused on driving these early and mid-stage clinical trials now into mid and late-stage clinical trials in the indications I mentioned, but also seek to commercialize through various transactions with big pharma that would keep us doing what we're really good at, which is developing these drugs into phase two, phase three assets, and then seeking a commercialization a partner for sales and marketing. And to that end, in the past 2 years, we have really been able to keep our heads down, execute against that. Because look, at the end of the day, whether you're AI drug discovery company or not, if you're a biotech, or the collective goal here is to get drugs to patients. And I- I think that is, at the end of the day, the most important metric.
The use of AI is becoming ubiquitous in drug discovery, whether you're a next generation AI drug developer or a traditional one. But how does your NI interrogative biology platform differ from what other AI driven drug discovery platforms may be doing today?
Yeah, so look, we've been able, being a first mover, we've been able to observe the development and the progression of this entire area. And I think what you've seen, to be frank, is a lot of noise in this area, right? There are many companies. There are a few great, companies that are doing great work along the lines of, looking at image analysis and trying to see how cells behave differently, like Recursion, or in silico, looking at, computational based, chemistries. But what really separates BPG and the NI interrogative platform is that in the first instance, we're interrogating, like, literally the biology of the disease. So firstly, we want to understand the disease. The other differentiator is that we actually seek to then develop the actual drug or a chemical that's going to go into human beings for testing. And we're agnostic to whatever modality. It may be a small molecule, maybe a biologic, uh, um, and we use very high throughput computational methods with a collaboration with the Oak Ridge National Labs. So as we're building out our biological models from these patients, we're able to put this on the world's fastest computer, at the Frontier computer at Oak Ridge. And in a matter of hours, we're screening 2 billion molecules and 2 billion, uh, certain like motifs or areas of proteins that may be a match for us to develop these drugs for humans. But thirdly, we actually perform the clinical trials and take these drugs into the clinic. So we're full circle from understanding the actual biological mechanism of the disease, developing the asset and taking that into clinical trials. But we're not just looking at a chemical based like computational approach and finding the molecule, and then we do and doing discovery or development on that, or finding a mechanism. We're actually fundamentally trying to beg the question: is there a specific gene protein pathway that's involved in this disease process? And the group of assets that we've amassed now that go beyond the AI, like having access to the Frontier computer at Oak Ridge, or like having 68 global relationships around the world with medical schools and hospitals that feed our biobank that's over 100,000, annotated like tissues and samples from that have helped us build 15 disease models. So it's the asset of having the compute power, having a biobank where you're starting with the tissues of the patients to generate hypotheses, and then taking that information and developing drugs in a way that's a lot more de-risked and triage. So by the time we get into clinical development, you want to do a few things. You really want to try to understand as much as you can how that drug works, like what is the mechanism. You want to be able to try to use the information to choose the groups of patients that it may have a higher likelihood of working on. And you want to then be able to plan specifically for the types of indications that you want to look at early on. But even in a late stage environment where we already are seeing activity in certain mitochondrial diseases or certain cancers, we don't stop there. Actually, this is a dynamic use of this platform where we may look at other indications. So the drug that's in cancer, like what other cancers are we predicting we may see activity on? So, using it for life cycle management of intellectual property, using it for to expand, you know, the market capture of a given indication that, for example, in cancer, but also using it to make sure that we're mapping and tracking efficacy and looking for safety signals in the most robust manner. So I think taking them all together, this is not only a discovery or a development platform. This is really an interrogative, like clinical a platform that helps us understand how these drugs are working and in live form in these patients.
So, would you contrast what you're doing to, say, identify a potential target to what a traditional drug developer is doing? Is would you say they're just being more hypothesis driven than you who have done the experiments?
Look, I think I would have said that 10 years ago, but I've seen also a tremendous adoption and a much more open mind and from even mid-size and big pharma. So I think it's our our real USP was that is that we're using patient data to create hypothesis instead of the hypothesis creating patient data. But more and more, the community, the entire industry now is recognized that at some level in most areas of drug development, we really do need to understand these diseases and allow, as you say, better hypotheses to be developed I do think that BPG has not only demonstrated a historical success point in that component, but we've continued to refine and extend the use of the platform from not only the wet laboratory or into preclinical studies, but now comfortably into clinical development. And that's really an advancement over over the years that's allowed us to span the entire process of discovery and development. And it's been tremendously successful for us for our own pipeline.
BPG Bio uses the term biology first to describe itself. It's a phrase I'm seeing more and more companies use these days. In some respect, I'd hope any drug developer was a biology first company, but is this just meant to say, yes, we're using AI, but that's not what's driving the science here?
Dan, look, I can't speak for others, but I can speak for why we chose to say it's biology first, because it is. That's We don't start any other process in this company until we have tissues and samples from subjects or from patients. So what we mean by that is that we're not just gonna take public data or look at existing hypotheses or try to enhance or improve or extend upon them or do a me too target based computational analysis. We actually have no upfront. We're agnostic towards how the data is gonna play. We're actually trying to follow the data. So when we say biology first, we mean that the first thing we're gonna do in any process is use the data from the biology of the patients, and whether that it means we're looking to discover like new drug targets in a given disease, or we're looking to drive certain like molecules or development around the chemistry, or we're looking to choose which patients we're going to put into our clinical trials. It always harkens back to that. So that's what we mean by biology first.
You made reference to your biobank. You do have an extensive biobank on which your AI platform performs multi-omic analysis. How unique is this, and what role does it play in the discovery process?
Well, it plays a tremendous role in the discovery process because it allows us, as I said earlier in this chat, was we really try to construct the narrative and the full picture of the biology on every single patient. And we believe, or look, the data demonstrates that we're much more than just the genomic data or proteomic data. We are complex systems, amazing systems as human beings. But we have like really important components that comprise key elements of our biology that drives really really simple processes, like how we break down glucose, right? It's like there's genes, there are proteins, there are lipids, there are metabolites, there are enzymes. And when you comprise doing like multi-omics, we want to look at how all of the various components of biology contribute to a signal or contribute to a process or contribute to uh, um, a mutation, for example. So if you have a gene mutation, great. So you sequence the patients, you see the patient has a mutation, and you can correlate that to oftentimes to a given disease. But is that the cause? Is a mutation the cause, or is the mutation a correlation? So in order to really truly understand that a patient has a mutation, which results in a protein product that's different than individuals who do not have that mutation, that then causes the production of certain metabolites and alterations of lipid signaling, such that the interaction both within the cell and between the cells, they then start to initiate a disease processes. So by understanding that entire milieu of information and the signaling cascade that is a result of mutation helps us to really understand the disease progression and process. It helps us look at potential biomarkers that we can leverage along the axes of that understanding. But it also helps us pretty rapidly in some cases to look at for example, a response or a toxicity marker in a metabolite or a lipid, because those change. Your those mutations aren't going to change, right? You're born with these mutations, or you have a higher risk. But the things that change in a dynamic and fluid manner are things like your protein translation products or your metabolites or lipids, and we're able to leverage those and tie them back to certain disease processes, and it allows us to just be a lot more robust and how we not only think of the disease process, but also develop solutions for it.
Well, walk us through the process of how you go about identifying a target and having a candidate that's ready to advance to the clinic. How much of that is done in silico? How much that is done in a wet lab? And how do you feed back experiments you do in a wet lab into the NICE system?
So the first thing that the BPG does as we think about how we build our models is, as we say, from a biology first perspective, we acquired the tissues and biofluids and the data from patients. So we acquire, for example, in pancreatic cancer, we you know we performed something called Project Survival, which we went to some of the leading hospitals and medical schools like Harvard Medical School and Mayo, etc. And we were able to acquire tissues both that were diseased and non-diseased from the same patient. We're able to get the pancreatic juice. We're able to get blood and urine over time, so that when we're collecting this amount of data and information, there's genetic data associated to those tissues and samples, and then those then come to our laboratories. So you ask how much of it is wet lab and done in house versus in silico. The other really differentiating factor, there is no use of public data in our models. Every single one of our models are built with data from the patients. So we're not taking like publicly available, genetic or protein data. They then come to our laboratories where we have um, a, a huge number of mass specs that are focused on doing in-house proteomics and metabolomics and lipidomics, and those tissues are subjected to a bit to a pretty robust like multi-omic profiling. So the only thing we don't do in-house is genomic sequencing because there's really no need to, right? It's so widely available. But our protocols are very robust around doing all the other omics internally. And then secondly, what we do from here is actually recreate an environment that mimics the cancer environment. So whether we're getting cells or tissues from these patients, we're able then to go into our wet labs and cancer doesn't like oxygen, so we put these cells, etc., in low levels of oxygen, or we use certain stimulators from the immune system, or certain components around the control of glucose. So we mimic these hallmarks of cancer, and then the omics is done all over again in those environments. So you're starting with the patient data, you're starting with the unperturbed expression of multi-omics with these patients. Then we then do something called perturbation studies, where we're perturbing these cells or tissues and putting them in environments that allow us to take the multiomics to see how proteins and how metabolites, how lipids are changing in these environments. Then all of that data is fed into a Bayesian AI system, and this is a really important differentiator. I didn't mention this earlier. So this is not generative AI. It's not neural AI, et cetera. This is specifically causal AI. So because what we're seeking to understand is, or what exactly are the factors in the biology, genes, proteins, et cetera, that are that we can say are causative. So we take the expression of the multiomic data in both a perturbed and unperturbed state. We have the patient's demographics, their clinical data, etc. And then in a population health perspective, we may build a model in hundreds or thousands of patients with match controls. So then it's exposed to the Bayesian AI system, and say if it's 500, patients with cancer versus 500 patients without cancer, all of the data is then compared. So you have the entire system and data in one bucket with the disease patients versus the non-disease. And then what we're really interested, Daniel, are the differences. So we overlay these two models, and then we build something called a delta model. And that delta model is essentially the changes within the system after all of these comparisons are done. And these are trillions of data points at this point, because you can imagine how much data is being created in these models. And then what results is a causal predictive model that looks like an airline map. The old airline maps that show American is hubbed in Miami or Dallas or Kennedy, etc. So you know that there's in those nodes and those central hubs and those central nodes, there's a lot of activity around the control of any operations of that airline. But in this case, those nodes also speak to control and changes between a disease and a healthy population. And that's really where we then use from the wet lab of the patient and the wet lab of all the systems biology and perturbations around the patients, go into an in silico dry lab environment. But we don't stop there. That's where we start. So then what we do is we go back into the wet lab, and we say, okay, well, if this our model predicted that a certain gene or protein has a causation to a certain cancer, well, we then use either CRISPR or siRNA, and we knock in or knock out that gene, and we see, okay, do you see a cancer cell now is going through cell death? Do you see that in the case of maybe it's diabetes, there's more like glucose uptake in skeletal muscle, or in neuronal models, you see that there's more mitochondrial activity that's indicative of a change. So that then results in a clear phenotypic validation that is disease relevant in the wet lab. It's, firstly in cells, and we also do animal models, and that then represents the validation, the validation set, um, the data set that is representative of a validated drug target. And then look, in some cases, we've even used the human tissue to validate. And the example is if you like in the cases of Parkinson's disease, where you're able to get fibroblasts, skin biopsies and skin fibroblasts from Parkinson's patients, and then you differentiate those into neurons, and then use CRISPR to knock in or knock out the predicted gene or protein target, and then you look at those functional studies. So we really pride ourselves on the robust validation processes, both within BPG, but mostly important within the laboratories and the environments of all our partners around the world. Because as we publish robustly with some of the leading institutions around the world where they're doing the independent validation, and we really do believe in that. So partners like the Department of Defense and the NIH and Oxford or Harvard or Stanford, these are institutions that we've had a long-standing relationship with, where that balance of internal versus external innovation has really allowed to the core demonstration and validation of not only the platform, but our early drug targets, and then of course, in the past like 5 to 6 years, our actual drugs.
How do you go about determining what indications you'll pursue, and how dependent is that on being able to get the biosamples you need to drive all
That's
this?
a great question. So look, I mean, we made a conscious commercial decision that we would focus on things that really, from a future perspective, when you look at the economic burden on not only the United States or the developed world, but what essentially is leading to the burden on healthcare. It's a huge increase in age-related cancers. And of course, now we're seeing even younger individuals, for example, younger women under 40 who've never smoked, that that are getting an increased rate of prostate cancer. So cancer was always going to be one of our core focus areas. Also, when you look at the epidemiology on brain health, uh, so aging populations with neurodegenerative diseases like Parkinson's and Alzheimer's, and then thirdly, inflammatory diseases, but more specifically, rare childhood inflammatory diseases, because unfortunately those are getting less and less rare. So that's really where the company's core focus has been, and collecting tissue. To your question, that's why I said it's a good question. Look, it's a little easier to amass the tissues and the acquisition in oncology because obviously there are more patients and they're more like centers. But in the cases of rare diseases, it does take longer, and you do have to sometimes even start clinical trials to collect those tissues or use drugs as you collect those tissues in rare diseases. And then in the neurosciences, uh, the nature of the collection of tissues around peripheral elements like skin or white blood cells, etc., um, has been has been a bit of a challenge over the years. But excitingly, we were part of the recently the Living Brain Project. So it is a unique element. This is why our biobank is so precious, and it is a massive differentiator to other companies in this space and other processes and other approaches to drug development, because we really do put a huge concentration on the interrogation of these tissues and cells and patient-derived cells.
Is there an example of a therapy you've advanced to the clinic that shows how your AI system was able to either accelerate development or do it more cost effectively or come up with a novel target, something unrecognized in a disease?
I'll give you two examples. So one is now in the subject of phase 2b trials for glioblastoma, and the same molecule has just ended phase 2 in pancreatic cancer. So for a long time, as we built our cancer models, so some of the core areas within the predictions, focused uh, on mitochondria and control mechanisms around the metabolic control of cancer. So we were able to take a molecule which for a long time has been viewed as just for energy production, ubidecrenone or CoQ10, and formulate that in a way based on the insights that we derived from the mechanism of action, formulate it in a way where we can make this into a pharmaceutical uh, drug formulation where we would mimic the elements of mitochondria, but then make that into an intravenous uh, nanodispersion. And that IV drug, now we took it into a trial of all comer solid tumors. We enrolled 104 patients, and we analyzed how this drug did in various cancers. Some were more. So the platform predicted that the drug would work in more highly metabolic, more aggressive solid tumors like pancreatic or GBM or hepatocellular or esophageal. And for the most part, that's what we saw, because we collected the tissues and samples on patients while they were alive in trial, and we built an AI model on every single patient in the phase 1. So we were able to not only understand the outcome from a response perspective, but we were able to assess the biological changes in those patients over time while they were using the drug. So then what we did at the end is we amassed all of that data on 104 patients and said, okay, of the patients who had stable disease or better as they used the drug, one, did it match the mechanism of action? And the answer to that was yes. It did. We did see the hallmark changes in mitochondria that would dictate that there were alterations in the tumor microenvironment and mitochondria to allow uh, the changes to allow the cancer cell to go through apoptosis again. Two, was it as predicted? Would it really work on more aggressive cancers? And the answer to that was also yes. When we compared the various cancer types, and then three, could we use that data to rationally then pivot into, as we did, into a phase 2 for cancers like GBM or sarcoma or pancreatic cancer, and we chose to do it in pancreatic and GBM. But then even as we did the further trials, the further phase 1s and phase 2s in GBM, for example, we did the same thing. So, but we also added in image analysis. So we would use 2D PET scans to demonstrate that these changes that we're seeing in the metabolism of the tumor was again reflective of this mechanism. So as we then further developed the drug that's now into a phase 2B trial, we've continuously validated and confirmed that indeed the drug was working by this mechanism. It also helped us to move from treating just the patients who were refractory. So in an early phase 1, the pool of patients that we had were patients who had already been given other treatments. So they had failed in some cases 3 to 4 different treatments. And as you know, Daniel, that causes a lot of resistant mechanisms and changes where, when you're coming behind any drug that's coming behind that type of resistance, it's always an uphill battle. But by providing the narrative on the patients, by providing the understanding of the mechanism, by demonstrating through pretty robust studies with Stanford on preclinically that if you gave the drug first line or front line prior to giving radiation therapy or temozolomide, that the results were much better. So what we did is we used the platform data, we used the output of the clinical mechanism, the analysis from the phase 1B trial, with the confirmatory studies that we saw in preclinically, and we took all this information and submitted for a frontline setting in a phase 2B trial. And I think without that volume of information and insights, it would be really tough to get that. But we were able to have that trial approved as a frontline therapy, and it's now been ongoing. It's probably it's past the mid stage of that trial, and we are seeing that not only is the drug continuing to be well tolerated, but we're seeing some really early promising signs of activity. So the earlier you get to the tumor, the earlier that you can start to make these changes in the tumor microenvironment and the mitochondria before they're subjected to resistant mechanisms, the better the better off this is. So I think that journey really highlights and underscores how important it is to take these approaches in drug development, because you can imagine that they're probably had we not taken this path, we may not either have been as successful as we are in this late stage, or it would have taken longer, or we would have not enrolled the right patients into these trials, which again, it's a huge trial and error. This is why even today, we we still have costs of drug development that's now up to $3 to $5 billion over 12 to 14 years.
The therapy is so interesting because it does target mitochondrial dysfunction, and by doing so, you're able to go after a set of indications that I think most people wouldn't see a relationship between. Is this working in the same mechanism of action across those diseases?
It's interesting. It is, and I think you make a really important point, because aside from potentially taking longer or taking a different route, you know, these insights into and as you know, there's a huge resurgence of interest and activity around the mitochondria as a targeting an area of research that has targeted for not only in cancer but other rare diseases, etc. But the mechanism specifically with BPM 31510, which is now has been studied in over 40 different types of cancers, but we're now really clued in on highly aggressive tumors like GBM and pancreatic, as I mentioned, and we're going to seek to expand this in other indications. What we're seeing is that the MOA is very specific to to tumors that have a higher rate of metabolism and tumors that have decided to, what's called a Warburg shift, where instead of using like glucose to break down as an energy source, they have made a decision to use lactate, which is, for example, when we work out or take a long run and you feel the aches and pains in the muscles, it's lactic acid production. But cancer also makes it a deal to say, look, I'll be less energy efficient. I'm not gonna use glucose as a fuel, but I'll rely more on lactic acid for production if I can evade apoptosis or cell death. And what this drug is doing is actually it's coming in and flipping the fuel in the cancer microenvironment, and it's doing so specifically in this class of cancers that has this mitochondrial dysfunction. And it's we've studied this now for almost a decade, from the early research to the preclinicals, and we see this across the board as a unified hallmark mechanism in cancers that exhibit this mitochondrial dysfunction. They they oftentimes are are a lot more aggressive than cancers without this dysfunction.
At the same time, does it give you an avenue to interrogate other diseases that might also have that same mitochondrial dysfunction at play?
So it has given us an opportunity to look at um, other rare diseases, other rare non-oncologic diseases. So we're currently also assessing an ultra rare disease called coenzyme Q10. I'm sorry, primary CoQ10 deficiency, which really results in a ravishing set of symptoms and uh, oftentimes very small children, but one can acquire some of these dysfunctions, based on your mutations over a, a lifetime, as sometimes as late as in your 20s or 30s, and it has allowed us to take the learnings that we've done and we've acquired in oncology and take the formulation approaches and essentially extend them into this rare disease setting. So, this is an example to your earlier question. Well, how's the platform really helped you to develop drugs or targets or mechanism of actions, etc. This is really an example where, because it allowed us to be so robust and understand so deeply how this drug, like, not only targets mitochondria, but also behaves and it helps to either abrogate or correct a dysfunction. Then you can imagine, well, okay, well, you're correcting this dysfunction and this is a cancer environment, But what happens if similar dysfunctions that are mitochondrially related are either abrogated or corrected? For example, in rare diseases or inflammatory diseases, or either metabolic or neurological diseases. And I think that with the um, you know I don't want to say advent, because it's certainly not an advent. Mitochondrial research has been around for a long time, but there's certainly a resurgence and a greater appreciation over the last 2 years, where we now are, uh, as a community, as a greater community, recognizing the importance of mitochondria as it relates to not only cancers, but neurodegenerative diseases, inflammatory diseases, both acute and chronic, and a list of other, you know, debilitating diseases that cause a lot of, not only disease burden, but economic burden. So, as you can imagine, we are looking at where the AI for us plays in a very dynamic manner because of our biobank, because of the tissues and the data that we have. When we're going to go build out our other rare disease, like secondary mitochondrial dysfunctions, we're not starting at zero, because we have a whole body of information from which to draw. But again, it's patient-centric information. So it really allows us to build out future pipelines and future drugs, and also de-risk the company's development in a really unique manner, because it's not just one mutation for one chemical for one indication or one disease, and that's it, right? So that's pretty binary. It's either going to work or not. And so, from a business perspective, this also helps us to uh, um, to reduce the risk in drug development.
It's been a difficult financing environment for biotechs. You're privately held. How are you thinking about raising capital, and particularly given the current financing environment, has it caused you to narrow your focus or reduce staffing or do anything differently?
So look, we've been keenly focused, and since the transaction with BPG Bio in January of 23. Thus far we've been able to really transact in ways that allowed us to perform the trials and resource the early R&D. For example, also on you asked about other areas in early research, like E2 biology, which is in the ubiquitin proteasome space. This is a really hot area of research. But in this, look, I don't think any CEO can tell you that it's not a tough, financing environment right now. I think it's probably going to get tougher before it's easier. But our focus has been on with the investment by Dan Elliott and his group. It's been more of a hybrid of a family office and private equity uh, perspective. That has been huge for us as an institution, because it's allowed us to not only focus, but it's allowed us to still have enough of a uh, an ability to resource the platform in building out the next generation of drugs. Because look, we're really excited about the phase twos and the near phase threes that we're focused on, but we also want to ensure that there's a constant development that's coming from the early research. So thus far, we've been extremely fortunate to be able to do that. But our approach now is from a financing perspective, as our data is maturing, with our phase two readouts and our clinical readouts. We have a lot of active discussions with strategic, potential partners in pharma, and I think that our our core focus is driving the highest quality clinical data, R&D data, so that we can secure those transactions. That, of course, would drive the revenue for us to continue to focus on building out the next generation of assets.
Niven Narain, CEO of BPG Bio. Niven, thanks as always.
Thank you, Daniel. Thank you so much.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.